Good afternoon, everybody. My name is Wayne Heacock. I'm one of Agilent's biopharma business development managers, and I'll be your host today. It's my privilege and honor to welcome our speaker, Harsha Gunawardena. Harsha got his PhD at Purdue University, where his research focused on advanced multi-source ion trap mass spectrometry with a focus on studying gas phase dissociation of large ions. He went on to do his postdoctoral fellowship at Lineberger Comprehensive Cancer Center at the University of North Carolina at Chapel Hill, where his research focused on large-scale quantitative proteomics. He was a co-investigator for the National Cancer Institute's Clinical Proteomics Tumor Analysis Consortium, CPTAC, and was a member of the UNC ENCODE consortium. He's currently a senior principal scientist and group leader of proteomics at Johnson & Johnson Innovative Medicines. Again, it's my pleasure to introduce Harsha. He's got a really excellent presentation today on some advanced workflows for utilizing analytical tools to advance biologics productivity.
Thank you, Wayne, and thank you for those kind words and nice introduction. So it is my great pleasure to be here today presenting our work that was a collaboration between Agilent Technologies, CMP Scientific, and Janssen, now known as J&J. And today I'll be talking to you about how we can couple mass spectrometry with non-mass spectrometry assays. And we've done this to automate profiling of antibody impurities. So I would give an outline here. First, I will show you why we need both speed and comprehensive characterization in pharmaceutical R&D. And my talk is two parts. First, I will go through a proposal for peak identification, a new strategy for identifying peaks in non-mass spectrometry assays. And here I will first describe what the peak detection problem is and give you a bit of a history of how folks have used new peak detection methods to characterize impurity profiles where there is no mass information available. In this section, I will go over three examples, case studies you can call them, and how we are using these studies to look at impurities using this approach. In the first, I will describe an approach where we are looking at hydrogen sulfide-induced degradation products of antibodies. And this is in itself an application in pharma where these degradation products are prevalent due to feed conditions, but we are using this as a way to do mass calibrations, and I'll get to that. And then we have two other case studies where we're looking at cathepsin-induced clipping, which mimic cell culture harvest conditions, and also high pH degradation products under post-deg studies. In this case, we're using rituximab as the case. In the second part of my talk, we are looking at data automation, and data automation specifically on the Protein Metrics Biosphere platform, where we're trying to bring in mass spectrometry data as well as non-mass spectrometry data in a seamless fashion to do the analysis. And in this section, I will describe you a workflow we've developed combining rapid fire for high-throughput mass spec characterization along with all the other non-mass spec assays. And the data analysis pipeline will be briefly discussed with how we automate the analysis for reporting of these impurities. And finally, conclusions and future perspectives, which are of mine. So, as we all know, I don't think I need to explain what a monoclonal antibody is to this audience, but there are about 150 such molecules that are currently being developed, that are currently commercially available, both for diagnostics and therapeutic purposes. But more recently, we've seen these bispecifics and also multi-specific antibodies that are used for both oncology as well as for targeting immunological targets. So So these antibodies, the bispecifics or the multispecifics, don't come without any problems. For analytical chemists, we can see that these problem areas or critical attributes have to be addressed, such as aggregation, chain mispairing, as well as clipping. And in today's presentation, I'll focus on the third point, which is clipping or degradation products that are generated in developing these antibodies. So just to give you an idea about where we sit in the R&D pipeline at J&J, I wanted to always put this slide up, is that we sit in between discovery and development. So you can see the discovery phases in blue and the development in red. And I come from the cell engineering group, which supports the analytical characterization of molecules that come from lead selection. So you can see that the number of molecules decrease as we come from pre-portfolio research where targets are declared. Then you have thousands of molecules which get funneled down to the single digits, and then it enters cell engineering. So during cell engineering, we are screening for the best clone that goes into late phase development. So no matter whether we are doing antibody engineering, where we're looking for the best molecule, or we are looking for the best clone, we believe that analytica cannot be the bottleneck. And so today I'm going to discuss some approaches we've taken to do both comprehensive analysis, but also increase the throughput. And we believe that one way to increase throughput is to rely on automation and how to automate these workflows, both analytical automation as well as data automation. So here's a pipeline that we've developed over several years now to look at both mass spec data as well as non-mass spec data. So the workflow starts with the LIMS system that collects and registers the data, and then it goes through this sample workstation in 2, where robotic sample preparation allows you to prepare all the samples. What's different about this robotic sample prep station is it is also interfaced to 2 mass spectrometers, and these robotic arms then allow the sample preparation to happen and data collection to happen in a seamless fashion. But we also know that this cannot be implemented throughout all the analytical devices, and we think that these mobile robots might be a better solution to do distributed robotics, and we've already have this in place right now to do the connection between the sample prep workstation to a non-mass spec assay that's shown here. So there could be non-mass spec assays as well as mass spec assays, but whatever data that is collected that goes stored into a data server with appropriate data. So if it's mass spec data, there'll be mass spectrometry data like spectra and so forth. If it's a chromatographic analysis, there'll be chromatograms or electrophoregrams that are stored in this data server. They can be physical servers or they can be in a cloud. And then in number 5, there's automation module that picks up the data from this server using a watcher and automatically uses the appropriate workflow in 6. And this all happens in the cloud and does the data processing. And now you can see the benefit of this is that we can connect different assays all in one place on this, using this cloud-based system. And once the data reports are generated, they are reported back to the stakeholders where validation of the data actually happens. But all the groundwork is done prior to this in complete automated fashion. And in 7, once the data is aggregated, they are reported back into gene data where it all started, where molecules get registered. And that's what we use as a data aggregation platform. So this is how we do the analysis, and the importance of this can be evident in the work that I'm going to describe to you later. So in this part, I'm going to show you the peak identification strategy and what I mean by the peak detection problem. So as you can see, we have a lot of these instruments in our labs which can do good quantitation. So you can have an absorbance measurement and you can quantify these peaks, but there's no way that we can identify if there is no a detection method to know its identity. So the ID piece is the missing puzzle. And in this work, we focused on mostly generating all the data on a chip-based electrophoretic system in GX2 which is now sold by Revity, and there are other devices that do the same thing. More recently, I've been told that there is the proteoanalyzer, which is multiple capillaries, 12 capillaries that can do capillary gel electrophoresis to separate different sizes of impurities in a very high throughput fashion. But in today's work, I'll be focusing on our work that we did with the GX2, lab-shipped GX2 system that's shown in the middle. So the motivation comes from this publication that we came across from 1966, which John Swan showed that the mobility shifts from SDS-based gel can be calibrated by taking the retention quotients plotted as a function of the log of the molecular weight. So if you do that, you can see nice linear calibration, and using this linear regression, they were able to identify or assign a mass to their unknown proteins. And taking this idea a bit further, our collaborators at CMP Scientific and Agilent Technologies, in a collaboration with the Chinese FDA, came up with this paper 2 years ago, where they used capillary isoelectric focusing as well as capillary zone electrophoresis to measure impurities in IgGs. And they were able to use this calibration approach to assign the identities of these impurities. So having these studies done earlier, we collaborated with both Agilent and CMP Scientific to study this more extensively in this study that we published last year, where we are generating a library of impurities using capillary zone electrophoresis coupled with mass spectrometry. And then we are trying to assign impurity peaks that are found in the GX2 lab chip-based system to assign peaks based on this spectral library. So I'll be talking about this study today, and I'll show you what we've come up with and how we are going to, use this study to further improve this workflow to fully automate it in the biosphere system, which will be in my second part of my talk. So the workflow is as follows. You start with your samples. They could be mAbs that are produced in bioreactors, or they can be also coming from formulation studies. And no matter what we do, antibodies can be subjected to degradation studies like forced deg, and you generate these impurity profiles. And that's what we did with standards, and this standard was used in this initial work, where we are first measuring the impurities using lab-shipped GX2, but we don't know the identities. But we take the same sample, and then we run it through CZEMS using Agilent's CE system coupled to the Agilent's 6545 XT QTOF system. And that will give us the masses of the impurities. And then we take it to the next stage, where we are generating the calibration using these standards, which is shown in the middle of the third panel, C. And once we establish the calibration curve, you can see at the bottom we also have identities of certain impurities that come from mass spectrometry. So those impurities then can be used through the calibration to assign peaks in the top electrophoregram in C that we get from the GX2 system. So finally, taking all this data, we can actually annotate peaks, especially unknown peaks. And you can see that in B, peak 2 was not identified before, but with the calibration approach, we can actually assign the peak. So this is the overall peak detection strategy that we've implemented to make use of mass spectrometry, assign peaks using a calibration. So we did 3 studies, as I mentioned earlier. So we have the first study, and all these studies emulate scenarios in bioprocessing. The first is where we are using an antibody, the NIST antibody, and we are subjecting into hydrogen sulfide-induced degradation, so incubated with hydrogen sulfide. And this actually mimics situations where certain feed conditions, such as cysteine, in a bioreactor causes hydrogen sulfides to reduce the antibody into its chains. So if we can take advantage of that, we can actually use those degradation products as known calibrants to do the calibration. In the second and the third cases, we are showing cathepsin degradation products, and cathepsin, as we know, is a wholesale protein that's secreted into the media, and these can induce various types of clips in your biotherapeutic. And the last example I want to show you is under forced degradation conditions as like high pH, you can monitor the fragmentation of certain antibodies that are susceptible to degradation, and I'll be showing you that case study as well. So this is the electrophoregram that you get after hydrogen sulfide, bubbling of hydrogen sulfide into a sample vessel. This can happen naturally in bioreactors as well, as I pointed out, due to the cysteine feeds or lack thereof. So there are differences in the amount of degradants that are produced. So you can see two peaks. One is when NIST is unstressed, and when you have hydrogen sulfide, you can see there is an increase in these peaks, but we don't know what they are, right? So, and also to note is that the time it takes to do this is about 30 seconds in this GX2 electrophoretic separation. So it's very rapid, it's a high throughput method, and the benefits of that are huge because we can process large number of samples. But if you look at the migration times, each second accounts for about a mass of about 15 kilodalton, which is huge, and that gives a huge margin of error. So the tolerance for error is very high, and this is the problem in the calibration. And what I want to point out here is the GX2 calibration that's done makes use of low molecular weight markers that are used as standards and to know unknowns that are high in mass, such as these antibodies and their fragments. So it's a mere extrapolation of the low molecular weight markers to look at these impurities, which is inaccurate due to the nonlinearity of this calibration. So the strategy is to see these peaks being assigned, and you can see that the GX2 peaks were assigned at the beginning, and we run the GX2, we run the mass spectrometry using capillary zone electrophoresis coupled with mass spec. We collect the data, and then we generate the calibration based off these mobility markers that we get. We call this the mobility ruler, where we are doing the same thing, plotting the log of the molecular weight with the inverse migration time, which is the mobility. And then we can, based off this calibration, we can calculate approximate molecular weights of our impurities. I say it's approximate because we've developed methods to refine these assignments, which I'll get to later on. But once we know what these molecular weights are, we can then annotate these peaks quite well. And this was all done manually, and I'll get to a point where I describe how we are trying to automate all these peak labeling while we are doing these assays. So once the peaks are identified, then the whole process starts all over again for the next sample and so forth. So this is a continuous monitoring process of impurities, combining mass spec with non-mass spec assays. So this is the mass spec data that we get from the Agilent CEMS analysis that we did for these standards, and you can see that we identified almost all the peaks, but except for the heavy peak that was not identified in this mass spec detection. And there is some, importance of this to note is that we don't need to detect all the peaks once we have a calibration, is that through the calibration we can detect, or you can estimate or infer is the better word, these unknowns which could potentially come from your calibrants. So taking this information, we can go and then make these assignments, and now we turn the x-axis, which was migration time, into a molecular weight scale, and we get very accurate molecular weights of our impurities, and with very low mass errors. So you can see the mass errors are very low, that you will never get if you use the GX2 calibration. So you can make these assignments, and in addition to that, the heavy peak was not assigned, not identified by mass spec, but based on the calibration, we can infer that this is the heavy peak in this electrophoregram. So that's the benefit of doing this calibration is complete annotation of your electrophoregrams. So to give a little bit of information or more details on this calibration is, we detect from unknowns approximate molecular weights through a calibration, right, with some error. But what we can do is making use of CZEMS data, which have accurate mass information from mass spectrometry, we can more precisely or more accurately get masses of your impurities. Another advancement that we made here is that we can actually use optimization methods to actually assign theoretical peaks that are possible with minimum time errors or minimum migration time errors. Both these greatly enhance our ability to make more accurate peak assignments, and I'll get to that in this next slide where we are using this minimization method. If you have a whole number of theoretical masses that are possible fragments, we can look at the migration time errors for the most suitable fragment that constitute a given peak. And we use this approach to make better assignments on our electrophoretic data. So in this section, I will describe cathepsin-induced degradation products of this antibody. And we are using two enzymes, cathepsin D and L, to monitor the degradation of this NIST monoclonal antibody. And we are performing CZEMS as well as GX2 analysis. And this is a very complex slide because this is an overlay of actually 6 electrophoregrams. So it's like 3 different conditions. Cathepsin D, which digests usually at pH 5, creates these traces in red. And pH 3 cathepsin L digestion creates these traces in blue, and cathepsin L at pH 7 creates these traces in green. I purposely overlaid them to show that they have different migration times. If I showed them separately, it would be hard to detect the time alignment of these different impurities. Now, if you want to annotate these peaks right now, we don't know what the identities are. So what we can say is that there are 2 time points. So there is a time-dependent increase in these peaks. And you can see that 2, 5, and 8 are due to cathepsin D pH 5 cleavages. Then you have the blue peaks, which is 1, 3, 6, and 9. And then you have the peaks in green that are cathepsin L, 4, 7, and 10. So as I mentioned, there is a time-dependent increase in the fragments. So this is all we have when we are detecting impurities, right? So we want to know the identities through the calibration that I just described. So what we found from the literature is that cathepsin L under 2 different pH conditions gives unique clips. So there is a AA cleavage site on the light chain of this NIST sequence, and a few residues downstream of that, you also have the FI clip. And those cleavages are very specific, and they happen at 2 different pH conditions. So one fragment, the AA clip, generates the 11.8 kilodalton fragment, and then the other one produces a 12.3 kilodalton fragment. But once it fragments, what's important to note is there is a complement mass of that fragment as well. So which is shown here is that you have the intact antibody, you have the fragment, which is a small piece, for this, in this case, the A clip, but there's also the complement mass of that fragment, which is also shown here. And we try to detect all these fragments in our electrophoregram. So how do we actually confirm that these fragments are happening is we do mass spectrometry using CZEMS, and this is the deconvoluted mass spectrum of both the AA and the FI clip. So what's important is that mass spectrometry can measure mass, but it can also confirm the mass by sequence, and we do a tandem mass spec experiment to mass select the charge state of this clip, which is on the left-hand side, and then we fragment it in the gas phase to generate these peaks, which is called the tandem MS spectrum, and these peaks then can be assigned to the sequence, which gives added confidence that these peaks are true and the true identities of the peaks through this sequence confirmation. So likewise, we have this for FI clip. Likewise, we have this also done for the AA clip, where we get sequence ions and accurate assignments of these to the sequence, which confirms unambiguously that this is the fragment. So we can potentially do this MSMS experiment for all the fragments that we detect in impurity profiling and building the spectral library. So the spectral library is not comprehensive, obviously, due to technical limitations of the fragmentation. So in some cases, we have complete fragmentation in MSMS data, but we can also have cases where we don't have MSMS, we only have a MS1 data, which is just the mass of the impurity, which is still good for this approach. So now taking that information, we try to assign through the calibration what these peaks are. So pH5 cathepsin D digestion, we don't see a reason that we would identify anything because we didn't actually look at the mass spec data for pH5 yet. So for pH3 cathepsin L, we can assign these peaks. You can see the AA clips quite well. Peak 6 is a single AA clip resulting from the heavy light fragment of the antibody. And peak 9 is 2 AA clips from the intact antibody. So they were very well assigned, and we believe that peak 3 is the light chain. And this was a tentative assignment, but you will know shortly that these tentative assignments can also be reassigned based on more mass spectral evidence. So at pH 7, you can see that now we see the FI clip in 7, where we have the heavy light with the FI clip. And then 2 other peaks, 4 and 10, we assign them to the half antibody and the heavy light species or the clip, respectively. So this is interesting because we don't get much information, and still peaks 1, 2, 5, and 8 are elusive. And because we did not get mass spec evidence for that. So now this is what the electrophotogram looks like up to now. So we go back and we use the migration time error minimization method that I just described on theoretical possibilities. So these are possible fragments that are possible for degradation, and we plot the migration time error, which could give both plus or minus in migration time, but we get the absolute value plotted as a function of these different fragment species that theoretically have accurate masses. And then we use the minimization approach to assign peaks 1, 2, 5, and 8, where the migration time is minimum to these species. And as you may know, that peak 10 was the heavy light species that we annotated previously, which also had the lowest migration time errors for the given masses surrounding it. So this is very accurate, and the heavy light species actually validates this approach for detecting unknowns. So taking this information, now we know what peaks 1, 2, 5, 8 are. We knew what peak 10 was already. We can go back and we can make these assignments. So now we have a better picture of what these peaks are based on this new assignment. Now, but is this picture complete, or is this still accurate? So maybe not, because I will show you how we can go and dig deeper into the actual mass spec data that we get from CZEMS to really find alternative masses that can make these assignments more accurate. So we found a FL clip, which is a cleavage that happens right after the hinged disulfides to generate this Fab prime 2 fragment, which is close to 97.6 kilodaltons. It's a pretty sizable fragment. And if you go and look at the electrophotogram, you can see that there was a peak corresponding to that here as an impurity that we didn't even assign previously. So you can also dig into these electrophotographs more closely with more mass spec information. Now let's go back to the peaks that we identified as 1, 2, 5, 8, which were tentatively assigned based on theoretical fragments. But now what we are trying to do is get actual mass spec evidence for them by CEMS. So we did get evidence for this clip, which we call a HT clip, which happens on the heavy chain slightly above the hinged disulfides, which is shown here. And this clip can happen on one side of the antibody or both sides, but what we see is one clip, one HT clip species, the small fragment. And we get mass spec evidence here is the raw spectrum on the left-hand side, and on the right-hand side is the deconvoluted spectrum of that, the clip. So the mass spec data is compelling to say that these impurities do exist, and if they do exist, those clips can be annotated in our electropherograms. So the HT clip can now be annotated, and if you look at migration time errors between a HT clip and a heavy chain, you can see that they are very close, but the HT clip has a lower migration time error compared to the heavy chain. So we reassign that peak with this HT clip. Likewise, we also detected 2 additional peaks. These are low molecular weight fragments, approximately 18.3 kilodaltons and 18.5 kilodaltons, and you can see the electropherograms on the top for these fragments, along with how the electropherogram looks like for the intact NIST antibody. And the bottom left shows the raw data of these species. By integrating across all these peaks, we can clearly see that the 2 impurities are there at 18.3 and 18.5, and we also see the intact NIST antibody. Now, this is a doublet, and keep in mind that we see this pattern also in our electrophoretic data, and I will show that next, where we originally thought that the cathepsin D digestion products, which showed a doublet as a FI clip, that was a tentative assignment, but with more evidence from mass spec data, we can actually reassign this to these 2 clips that are shown here. So how about the heavy peak, right? So that's peak number 5, which tentatively is a heavy, but we find this clip, which is the SS clip. Between SS, the 2 serines on the light chain, there's a clip, and it can happen on both light chains to give to this heavy chain with 2 pieces, like 2 wings on either side. And this is very accurately determined, and the mass accuracy is good. And then we use this mass information and then go back and re-annotate it, and we find that this heavy was actually this antibody minus the 2 SS clips. So now we have all the impurities annotated on this electropherogram. And this is the evidence that we have of this reassignment in the migration time minima that we get through this analysis. You can see that peak 2 was assigned from the FI clip to these 2 doublets in 18.3 kilodaltons and 18.5 kilodaltons. And then we have the peak 4 reassigned from being a heavy chain to the HT clip, and peak 5 reassigned from the heavy chain dimer to the antibody minus the 2 SS clips. So now we have all the peaks quite accurately annotated, but we were never able to identify the true identities of those doublets, 18.3 and 18.5 peaks. We measured their masses, but we were unable to get convincing data to really sequence the ions in an MS spectrum. So we believe that with additional or complementary dissociation methods in the form of EXD or electron capture dissociation, which is now available on the back end of the collision cell of this instrument, we can perform these EXD experiments to get extensive sequence coverage of these fragments and to unambiguously assign sequence ions. Here is the enhanced ECD efficiency that's been shown for this peptide. What's important to note here is this peptide has 5 leucine and isoleucine, and as you know that these are isomers. And based on the fragmentation patterns, we can differentiate leucines from isoleucines using electron capture dissociation. and we can tell the difference based on the fragment ions. So that's the added benefit of doing tandem mass spectrometry with electron capture dissociation. So in addition to that, you may also know this sequence has aspartic acid residue, and these aspartic acid residues also have isoaspartate isomers, and through the fragmentation, you can also distinguish aspartate from isoaspartate using ECD. So the next case study I want to share with you real briefly is this high pH induced degradation products that are normally taking place. And we do forced degradation studies, and this is a good case for this where high pH causes rituximab fragmentation. What's special about rituximab is in the literature we found this nice paper where it describes the mechanism where ammonia in these buffers causes certain residues, especially the asparagine proline bonds, to be weakened and fragmented. So if you look at the rituximab light chain, there is an NPP motif which can potentially be clipped under high pH stress. And we were expecting this to be a good use case for us to go and investigate electrophoregrams under forced degradation studies of rituximab. So we did just that, and we first collected the MS data using CZEMS, and we actually find this clip. There's an accurate mass for this clip in the spectrum to your left. And then we collected the GX2 electrophoregrams, and we did this for the case where it was unstressed, rituximab at 4C, and then we also had high pH stress for 1 day at 4C, and high pH stress at 37 degrees. We elevated the temperature for a day, and then we looked at the peaks, and you can see that certain peaks increase in absorbance, fluorescence absorbance, and the degradance of these species increase. And now we can use that calibration approach I just described and make assignments, and we are looking at the NP clip mass, and we can make these assignments quite well, and you can see that the NP clip itself was found in peak 1, but we also saw the complement of that mass in the form of a single NP clip in peak 7, as well as 2 NP clips in peak 8 being accurately assigned. What we could not assign is peak number 5. So there is some limitations. If you don't have identity in mass spectrometry data, you might not be able to assign them quite accurately, even though you have accurate calibration. So in this part, I want to describe how we've automated this. So this was not in our paper, and this is current work that's ongoing, where we are trying to not just limit it to GX2, but we are trying to expand it to other types of non-mass spectrometry assays, as well as other forms of mass spec data collection methods. And here I want to make a case for rapid-fire MS, because rapid-fire MS is a very fast way to get masses. Every 10 seconds you can get a mass, and non-mass spec assays can be high throughput or low throughput, but the benefit of doing high throughput is that we can probably do real-time assignments them through this software in Biosphere. So we're building the pipeline capabilities to integrate these different types of data. So here I'm showing you the rapid-fire run of a 96-well plate. So what you're showing in the middle is a mega file of the chromatographic traces. That's not a chromatogram, it's just an injection traces with a few blanks in between, the washers in between. But each deflection that you see in the middle is an acquisition of a mass spectrum. And we can do that every 10 to 12 seconds. And we can complete a 96-well plate with no washers in 20 minutes, and with washers in between, we can do from anywhere from 30 minutes to about 40 minutes. And once the data is collected, it's automatically split into individual files and sent to this Biosphere automation system, where the data is processed. So this is the pipeline I described at the very beginning, and it generates reports. And what's shown on the right-hand side is the Biosphere plate map report. So what it's showing is the exact layout of the 96 well plate as a report with different colors. So you may know what these colors mean. It's green, yellow, and red, just like the traffic lights. Where things pass are green, and where things are ambiguous, it's yellow, and if it fails, it's red. And this pass, fail, and maybe kind of annotations are given based on the identities of the known molecule, which is an antibody, as well as if they are known along with impurities, it's a maybe, and if there are impurities without the intended antibody, it's a no-go or it's a fail. So what you're showing here is a spectrum, a raw spectrum on the left-hand side of an antibody that had an impurity, which was a half antibody. And the deconvoluted spectrum shows the exact mass all reported within biosphere. So you can go and dig into any one of these spots and look at the spectra and inspect it manually. But since it's a high-throughput method, using this biosphere automation, we are able to generate these reports for users to quickly go and make decisions on the data, whether it's a good molecule or it's a bad molecule, and if so, what made it to be bad, whether there was impurities or not, and so forth. So this is a case where we had a half antibody, and this is another case where we detect the intact antibody, and this was a spot where we had a correct assignment of the molecular weight of this antibody. So this is the pipeline that we're building around this, is a complete end-to-end automation workline. This is still work in progress with protein metrics, or now dotmatics, where we are trying to look at all these different well plates coming from bioreactors, AMBER 15 or AMBER 250 bioreactors. We get several dozen plates, and then these plates can be run on any type of assay in the lab, whether it be GX2 or it can be anything like the proteoanalyzer from Agilent or anything that will collect very rapidly data. So in this case, it was a 40-second sample injection on GX2, and the same sample plate, a daughter plate, can be sent over to the rapid fire to collect mass spec data. Every 10 seconds, we have a sample acquisition. And because of the data collection is very fast in both these methods, it's really suitable for automation because it can be automatically swept into this vendor-agnostic automation module that will process both data simultaneously and jointly report information from both data streams. So that's the future goal that we have. It's still work in progress, and once the data is generated, it can be put in dashboards or it can be reported to the end users. So dashboards would be logically the obvious way to monitor these things in real time, but the reporting options is also useful because we don't need to do it as fast as seeing things on dashboards. So I'll just show you what we're hoping we'll get out of this. The data has been already collected, but we still do not have the biosphere in full operation to look at the data. So this is what we would imagine this would do, is a complete annotation of peaks in these electropherograms using mass spectrometry data. So we run the rapid fire, collect the masses, generate a spectral library of impurities, and then those are reported as these traffic light reports. And then that information is also shared with other assays to annotate peaks. And we hope that there's a molecular glyph that can also label each of these peaks so that users can go and take a look at this data and, with no interpretation, consume the data. So with that, I would like to conclude my talk, and I hope I was able to convince you this approach as a successful way to look at, and a radical, in fact, it's a radical approach to look at and accurately identify peaks in cases where mass spec is not an option, and there are either due to technical challenges where a hyphenation technique is not available with the assay that you have, and due to technical difficulties. And we can take this hybrid approach to bring in identifications from mass spec that was collected using a different instrument to the non-mass spec assay. I showed you several case studies that we did and how we handle impurities in a biopharmaceutical setting. We also looked at how GX2 peaks can be greatly enhanced by this approach, and we are currently working on rapid-fire MS identifications to look at non-mass spec assays. Now, there are some potential applications like use ECD as alternative fragmentation to look at IDs, especially in kind of a QC setting where molecular weight accuracy and identification accuracy is very critical, and we believe that EXD is a way to go and interrogate these unknown peaks even further. And we envision that this software will completely automate, and we'll be able to do seamless peak identification of any type of non-mass spec assay. So with that, I'd like to thank all my colleagues at J&J, especially the biophysical separation team, Colleen and Ben, who are very much supportive and instrumental in generating all the electrophoretic data. I'd like to thank the mass spectrometry team for their support, and most of the mass spectrometry data that was collected for this project was performed at Agilent and CMP Scientific, where they had the CZEMS instrumentation, and I'd like to thank John Sawson, especially for coordinating all the activities, as well as James Shah from CMP Scientific for this wonderful collaboration. They were great collaborators. And also I'd like to give a shout out to Protein Metrics, who have been really, really wonderful collaborators. We've been working with them for the past 6 to 7 years on automating our workflows, and if not for them, none of this would be possible. So thank you once again, and I would be happy to take any questions.
I should note that there's a couple of microphones in the center aisle there, so if anyone has a question, feel free to come up and ask. Thanks.
Yeah, it's very nice talk, and I want to ask, when you build the calibration curve, we saw you build the curve all the way above like 500 kilodalton, but all your target is actually below 150 kilodalton. Is there any potential to just focus on the low mass region to build a calibration curve?
Yeah, this is, so it depends on how, what the range is for the calibrants, right? So in Revity, which was the GX2, which was formerly PerkinElmer, the drawback to the calibration was we're using low molecular weight markers to extrapolate to higher molecular weight species such as antibodies and antibody impurities. So I would imagine that if you use a low molecular weight standard, and they got different types of standards, you can definitely look at the low molecular weight range more accurately. So in the case of antibodies, we are definitely wanting to go up in mass, not down in mass.
Thank you.
Yep.
I have a question. So for the interface of the CGEs with your QTOF, the Agilent QTOF, so the interface is made by Agilent or the CMP Scientific?
Yeah, this particular interface we use for CMP Scientific is a sheath flow interface, and they're probably working on other types of interfaces, but the one that, the data that I showed today was all from CMP Scientific.
Okay, great. Thank you.
Yeah.
We actually have a question online from Xiaoping He. It says, hi Harsha, for your first example, would you clarify if the GX2 CZEMS was unable to identify the mass for the HH fragment, or you just want to show the capability of inferred calculation of all the fragments?
Yeah, I don't know whether I understand it very well, but I heard HH fragment. Yeah, so the initial assignment of the HH fragment, heavy was not really identified by mass spec. Detecting, the heavy chain is somewhat problematic, and if people here do LC-MS, sometimes the heavy chain is sometimes not present in some antibodies by reverse phase as well. But in the case of CEMS, we did not detect the heavy chain, but we detected an alternative fragment, which was the SS clip on the light chain. And we have two SS clips that result in the complement mass of the fragment complement to the mass of the SS clip, which corresponds more closely than a H fragment. So since there is mass spec evidence for that, the impurity is most likely that of what we confirm by mass spectrometry, not what we inferred based on the calibration. So there's a whole lot of calibration being used to infer things, but we also have accurate mass from mass spectrometry really confirm the impurity profiles to really make peak assignments. So I hope I was able to answer that question.
There's a follow-up question too.
Yeah.
In the third case study, was the rituximab light chain clip specifically induced by ammonium or any other high pH environments? And also, have you seen that light chain clip in other mAbs?
So the light chain, so NPP motif is not too common. We don't see this in rituximab. It's a good use case for this. So we don't necessarily see, NPP clip, the motif, is important in the clipping. So we haven't done a whole lot of testing on other antibodies that have NP motifs to see if there are these particular clips, but this we found in the literature, and we tested it, and we wanted to make a case for GX2 peak assignments based on that prior knowledge. So there could be other antibodies that have similar motifs that we could test, and anybody can test it as well.
Hi, Harsha. Thanks for the talk. So you're using like a high-resolution mass spectrometer, and the GX2 is obviously lower resolution, right? Did you ever come across an instance where you found more impurities in your mass spec than you did in the GX2, and you had like co-migration in the GX2? And I'm kind of wondering like how you might handle that in your calibration.
Yeah, that's an interesting question. That's exactly true. I mean, because the GX2 is so high throughput, our run times are very short, 45 seconds, 30 to 45 seconds. You may have co-migration, and you may not have adequate resolution to look at certain impurities that you would otherwise get from mass spectrometry with a long CEMS run, right? And that's definitely a drawback. But if we were to implement an interface, so to speak, between a GX2 assay, for example, it's just an example, is how well can we do to assign peaks in GX2? Obviously, there could be co-migration, and there may be peaks that we don't detect completely, or we may miss things. But the idea is to make a complete case or complete annotation of the species that we detect in our GX2 profile. But that also brings interesting argument in terms of quantitation. If there was co-migration, could potentially, if we do systematic studies, we can see peak interferences, right? And the peak interferences can be evaluated because GX2 could be pretty quantitative with fluorescence measurements. And I think that might help to answer your questions about if there is some impurity buried in a peak that is not resolved or partially resolved. And even the electrophotographs that I showed to you today may have other species buried in them that we don't know because there is not enough resolution for that. So this is where, complementary methods will come in. I mean, we can do CSDS analysis with long capillaries at longer run times to improve the resolution of these unknowns. So, yeah, that's a good question.
Oh, I think we have one more. Thanks, Harsha. Xiaoping is happy with the answer.
Okay, I'm glad that he's happy with that.
Yeah. Are there any other questions then? Well, I would like to thank everybody for attending the talk. If you have any questions about the hardware or other software workflows that were associated with that, you can feel free to reach out to Harsha, myself, our colleagues at Protein Metrics. Thanks.